and welcome back. This is the last talk in our current series. Thank you for bearing with us. This is really exciting. Certainly for me as a doctor, this talk is really exciting as we're going to talk about neutrophil extracellular traps, NETs, being a potential therapeutic target. And I'm really just going to quickly flag to you two studies. Essentially the question is, we know that there's a strong signal towards severity of illness with NETs. A high NET level equals badness. Can you do anything about it? Looks like the answer to that question is a very definite yes. The first paper I just wanted to highlight to you was published by Garcia in late 2023 from Professor Jean-Louis Vincent and Professor Fabio Tacconi's laboratory. And it's looking at neutralization of extracellular histones using sodium beta oxymethylsilobioside sulfate in a model of septic shock. Forevermore, we shall call that MCBSS, as it's too much of a mouthful to say out loud. In their laboratory, they have a very established animal model of sepsis, where they use sepsis in sheep. And in this model, they inject feces into the abdominal cavity of sheep to stimulate sepsis and immune response. There were 24 animals involved in this study. There was a control group. They had the injection, and then they were monitored. And after 8 hours, they were given fluids, antibiotics, and treatment as you would normally expect. The second group was called a late treatment group. After 8 hours, they were given all the same treatments as the control group, but they had this molecule MCBSS added in too. And then, in a slightly non-physiological way, in that you'd never give a treatment as soon as you infect someone, they gave the last 8 animals this molecule MCBSS immediately after they'd injected the feces into their peritoneum. So really saying up front, can they see a difference in the response into these animals? Yes, they could. Of the animals in the control group, those animals who just got resuscitated with fluids, antibiotics after 8 hours, 2 of them died. 2 of them died before they got to the end of the experiment. They had increased markers of organ distress. They had increased serum lactate compared to the other animals, and they had increased markers of cellular distress. So they had increased markers of renal failure. They had a decreased number of platelets, suggesting their platelets were being consumed and activated. They had decreased urine output, and they had increased levels of interleukin 6, interleukin 10, which are key inflammatory molecules. Compared to the intervention groups, those who got this molecule antagonizing NETs and antagonizing the components of NETs histones, the animals in the control group had much higher levels of circulating H3.1. It's very early to say. It's clearly an animal model. It's not a human model. But it looks like antagonizing by lowering the levels of H3.1, you had a biological effect. You had slightly less severe illness, less evidence of organ dysfunction, and less dysregulation in these animals. That's a really exciting therapeutic window. It means you can give a drug to attenuate or to manipulate one of the components of NETs. And so can you dampen down, can you manipulate an excessive immune response? And that's a key therapeutic window there that we're looking at. The next slide is from our colleagues at Therapeutic, and this uses a column. Blood is taken outside of the body, washed through the column and returned to the body. And this column is excellent at removing H3.1. Indeed, the graph on the right shows that the columns are extremely effective at removing H3.1. They remove all the H3.1 in the system. What did that mean to the animals? There were 6 pigs in this experiment. The pigs had less lactate, this marker of cellular distress. They required less norepinephrine to support their blood pressure, and they had a better urine output. So what do these studies show? These are 2 studies in the real world, in animals with real world models of sepsis, showing that you can improve organ function, or you have an effect on organ dysfunction if you block the excessive action of H3.1. That makes it a really potentially exciting therapeutic target. It is clearly very early days, and we are some way from taking this to humans, but you'd start to see a pathway evolving in front of us. We now have a test that works. The test itself takes 15 minutes to run now, and as I said before, I'm extremely grateful to the work the research and development team have done in Belgium to make that possible. That's a real-world test that doctors could use. It's reproducible, reliable, and it's CE marked. We're understanding the science and biology of H3.1 much better, and we are identifying ways in which the chromatin unwinds, and we're starting to take steps towards, can we differentiate, can we spot the difference between suicidal vital and mitochondrial netosis. And these studies in animals are showing that there's a biological effect. And our studies in humans, where it's been observed, those levels have been observed in humans, really suggest that there is clinical significance to it as a marker of severity of illness and a marker to track a patient's progress. This final slide is just a start. It's very complicated, this slide. I'm not going to mention all the drugs in it, but there is a plethora, a huge number of targets that we could potentially consider manipulating, changing, adapting, adjusting to try and improve outcomes when patients have increased H3.1 levels. And so we're really excited. We think this is a really rich field for further research and further development. But if we take a step back, the reason I came to work for Volition is to bring a high volume, low cost test to market. H3.1 is a rapidly usable test, low cost, high throughput, to help clinicians across the world identify the severity of sepsis and track sepsis. I think it's extremely exciting. I hope you've enjoyed these series of lectures. If you're interested in more information, all the published papers involving H3.1 discussed today are available through our website. We've got a large stream of clinical studies and more scientific research coming. As soon as they are published and available, we'll record videos and report them on our website too. Thank you.